Thanks for the opportunity to speak today. I wanted to start with this slide. It's based on a series of papers that came out in 2015 and describes basically the budget surrounding, sorry, let me turn this off here. That described the budget surrounding the money that's spent in research, in life science research. There's about $114 billion spent in research, life science research. Yeah, I had this set to go into mode that's automatic here. So, and it's split almost equally between clinical and preclinical research. You'd figure that it would be split more heavily towards clinical, but because of the sheer number of studies that are preclinical, there's actually more that is spent in clinical research. I'm just going to do this for now, and then we'll go back the other way. Sorry about that. They found, and it's kind of astounding, that about 50% of the amount of money that's spent in preclinical research, about $28 billion, is not reproducible. And there's about 75% bias in all of those studies, and some of the results are about 27% over-representative. What are the sources of those issues? Some of them, it's most likely not intentional. I mean, there are some PIs that will align results to match some of their hypotheses, and I'm sure no one in this room does that. But I think most of it's unintentional. You could have reagents that aren't active. You could have protocols that aren't consistent across experiments. You can have study designs that are flawed in some way. We can't solve all of those problems, but we attempted to create a platform that will address some of the data analysis and reporting biases that you might have in research. So this is an example of an image that someone might analyze. It's a set of fibrils that are in a reticulin-stained tissue section. The state of the art is still to have somebody do manual analysis of this. The brain and the human eye are very powerful computer, much more than an algorithm can be, but it's also very inconsistent. It's dependent on experience, right? So what kind of who will do this kind of analysis? Well, you end up with this guy, right? He's going to be sitting under a microscope, clicking and counting objects, right? That it's not very efficient. And honestly, I was that guy a few years ago, and it's the reason why I started ImageIQ. It's not accurate. It's not reproducible. Depending on how many cups of coffee he's had, what his level of experience is, whether it's at the beginning of the day or the end of the day, it can be very, you can get different results. It can be biased if they know what the actual groups are. In some small labs, they will actually know what the outcomes should, or what the treatment groups are. That will lead to unintentional bias and counting more cells in one, or more fibers in one group versus another. It's a poor resource allocation methodology, because usually we recruit these postdocs and grad students that are talented for other reasons, not necessarily to count objects in an image. And it also creates access issues. So if they're sitting under a microscope counting fibrils, they're occupying the microscope. There are other options for this. So you can use commercial-based analysis packages. Some of them are here on the side here, so you can use ImagePro, Definiens, Velocity. Some of these are expensive. I think you can get Definiens or MATLAB up to like $20,000, depending on how many packages you buy. You can use open source tools, but there's a large learning curve for those too. What happens with these packages are to account for all of the variability in all of research, they add more and more filters in them. So you end up getting this bloated piece of software, and you can choose among any one of those filters to process your image, but it's sort of the Wild West still. You're able to adjust thresholds, you're able to choose different filters with different kernels, and each one will give you a different result, and it's not necessarily a controlled process. You could have one graduate student create a workflow pipeline for image analysis one way, and then it could be applied differently by another graduate student, and you'll get a whole new set of results. The other issue is you create an image specialist in your lab that really knows how to use all of this software, but if they end up leaving or there's a turnaround, then you have this piece of software that no one else can use and data that you can't reproduce. So, and I mentioned that it's hard to sort of version this sort of analysis here. So what we did was we developed a tool, a platform called imagequantify.com. And sort of the idea behind this is to actually create image analysis on the web. We chose Amazon as our sort of provider for doing this. There's no learning curve required. There's built-in version control. So when you use a particular IQ bot, and I'll go into what that is, it's maintained for you, and you can use it today, or you can use it 10 years from now. So there's no purchase of expensive software or computing. You don't need a specialized person to learn how to use it. All the algorithms are validated by pathologists, radiologists, depending on the assay that you're using, or just expert observers. And ultimately, the idea is to sort of reduce bias. So how does it work? It's just like if you've used some of these online photo album sites where you upload your images. You upload your images to an album, and then you choose something called an IQ bot. An IQ bot is essentially what we call these specialized algorithms for a given assay. So if you're doing tube formation or you're doing muscle fiber analysis, you'll choose that bot. And then you basically say click, you click a button, and you automatically, within about, depending on how complicated the algorithm is, an hour to 24 hours, you get back an email that says your data is ready. You get back segmentation overlays that show you where the algorithm measured or selected your objects or segmented your objects. You get data sheets with all of the quantitative metrics in it. You get methods and validation reports that show how the actual algorithm is validated and the specific filters that were used in it. We try not to hide those things. You can cut and paste them and put them into your publication or your grant. These are just an example of some of the IQ bots that are available. Anything from DAB analysis, quantifying color in your images, wound healing, bone ingrowth. If you go to our site, you don't see something that you are working on currently, you can just submit your images, and we'll create an IQ bot that's specific for you, and you can use it. Going back to that image that you saw, there was actually around 2,000 fibrils in that image. The algorithm that we created will analyze that image in less than 2 seconds. And what you get is data that is pretty comprehensive. So that guy that was counting the muscle fibers with his clicker is not analyzing how big those fibers are, but how round they are, what the aspect ratio is. The algorithms that are running do all of that, and then they give you these massive Excel sheets that you can then plot out as histograms, which has way more value than just counting how many fibrils you have. Because a lot of these trends that you see in pathology aren't very distinct when you just look at means, right? And the same thing for other assays that we created. So we do an assay for cell proximity. We had a PI that wanted to know in his glioblastoma cross-sections how close are his SOX2 positive cells to a vessel. So he labeled his vessels with smooth methyl cell actin, had DAPI for all of his nuclei, and he had SOX2 labeled with a marker. And we basically had the algorithm trained to pick up where all of these objects were with respect to the boundary of a vessel. And he was able to clearly see in different treatment groups where the SOX2 positive cells were located with respect to a vessel boundary. I can go on for the next 2 or 3 hours on how the different types of algorithms that we've written for these assays, but I'll go through a couple. Like tube formation is pretty commonly done in Matrigel, so we have a lot of companies and groups that are using that particular assay to look at connectivity and nodal networks within these endothelial cells in Matrigel. Astrocytes, counting the number of astrocytes all the way from just looking at the intensity of the cell body to the number of dendrites, number of axons, the branching of axons, multiple channels with different antibodies, and looking at co-localization. Transfection efficiency is a pretty big one, looking to see where, how many cells are just positively transfected within a big culture. Bone contact, bone ingrowth studies. This is an example of a graft study that they wanted to analyze where cortical bone was, where woven bone is, where graft material is, where connective tissue is. Algorithm runs, and this particular image was a large field of view image from a slide scanning system, so it's about 2 or 3 gigabytes, and it will run through and can analyze all of that within a few seconds. Fish, there's a lot of controversy on different fish kits and the results. So we have algorithms that actually can identify particles within cells, not necessarily based on thresholding. It uses other filters to actually pick those objects up. And all of these algorithms can get all extended to 3 dimensions. So if you're using micro CT, micro MRI, PET, the algorithms extend to 3D. You can look at connectivity. This particular example was for a rat lung that was infused with microfilm. So you essentially get all of the vessels filled with a contrast that shows up on a micro CT. You can then do thickness analysis, which is what you're seeing on the left here. And on the right, you can do some more sophisticated analysis, such as connectivity. So in this particular example, the algorithm finds the trunk and then all of the branches that are coming off the trunk. It looks to see every single bifurcation. And if you look at the image on the left, you can see that it's different. On the analysis on the right, there's an actual a fifth branching level event that's completely missing. So again, it's fully automated. You just give it the volume and it will run and crunch and do its thing. So again, I have a lot of different examples, but if you have any specific questions, you can visit us at booth 606.